Now we're going to talk about principles of inheritance, and we're going to discuss principles of inheritance by talking about Punnett squares. Now, if you've ever done a Punnett square before, or had any kind of genetics instruction before today, then you already know a little bit about this. You already know that it's an imperfect system. Not every principle of inheritance follows the 50-50 chance rule. We do have things like incomplete dominance. We do have codominance. We do have polygenetic attributes that rely on the interactions between multiple genes. The Punnett square is sort of the simplified version. The reason we use it and the reason it applies in biological and pre-health fields is that a lot of disorders will follow these principles of inheritance. We do have the concept of dominant disorders, recessive disorders, and the inheritance patterns that go with that do come through the statistical analysis. And what we are talking about for the uninitiated is a statistical analysis of what is the probability that any given offspring will have a certain combination of their parents' chromosomes. And this is very tied into the idea of meiosis that hopefully you just watched right before this video. So remember, in meiosis, you have crossing over, you have random assortment, you have a gamete that has a subset of chromosomes from the parental gamete. Right? So you started out diploid, you ended up haploid, so you cut the number of chromosomes in half, and it's a total 50-50 flip of the coin, whether it was the matrilineal line or the patrilineal line that ended up in that gamete. So that 50-50 flip of the coin is represented with this box right here. So this biologically female person with XX chromosomes was able to create some oocytes that were haploid, and randomly this oocyte contains the matrilineal line and this oocyte contains the patrilineal line. Different oocytes, because remember, meiosis means that you only have one cell at the end of it. Or another way to think of it is there was just a 50-50 chance that she developed this oocyte, or a 50-50 chance that she developed that oocyte. And those are homologous chromosomes. Those are not the same exact set of instructions, but they are the sets of instructions that code for the same concepts. So like eye color, right? Maybe this oocyte contains instructions for brown eyes, maybe this oocyte contains instructions for blue eyes. 50-50 toss of the coin. Meanwhile, the same thing was happening in the male system. 2 rounds of meiosis means that we ended up with haploid cells, and it's completely random, whether it's the matrilineal or patrilineal chromosome that ended up in any given gamete. So we'll put one parental figure on this axis, and we'll put one parental figure on the other axis, and we're going to cross multiply. So what I mean by cross multiplication is that we'll take these boxes, and again, this is just a statistical analysis. We'll say in this box, we will take this probability and combine it with this probability. So this is the likelihood that we'll develop an offspring with XX chromosome. And in this box, we'll look at another quadrant of probability. We'll combine this probability with this probability, and that's an XY individual. In this box, we'll consider these 2 probabilities, and in this box, we'll consider these 2 probabilities, like a cross multiplication table. You can do this with like branching Y patterns instead. I'm used to Punnett squares, so I do Punnett squares. So 50-50 chance that this person will end up with female offspring and 50-50 chance male offspring. And that's pretty much universally true, but it also applies to every other autosome, all of the other chromosomes. So the 23rd is the X or Y chromosome. 1 through 22 are called autosomes. That's every other chromosome. That is not about sexual differentiation. One thing I want to convey here is this is probabilities. These are typical outcomes. These are averages. There's not two biological sexes. There are other possibilities here. There is XXY, there is XYY, there is X0. There are, let's see, I think it's possible to get 3 X chromosomes. It's rare, but it's possible. So there's no two biological sexes. That was never accurate. And then gender is a completely different question. Gender is an identity, and this is a biological sciences class. And while we could get into some very advanced neuroscience and talk about why gender identity is more in keeping with people's brains. So people's brains are more like their gender identity than with their body's biology. We could do that, but that's not the discussion for today. So if anybody one-on-one wants to talk to me about gender identity, we can actually absolutely do that. But as a biologist, as a neuroscientist, as a neuroanatomist, as an anatomist, I'm going to go ahead and say there are more than 2 genders and there are more than 2 biological sexes. Absolutely. Anybody who told you otherwise has an agenda. Which is true. A lot of textbooks come from Texas, which has a political agenda. They have actually a bit of a monopoly in the K-12 textbook game, and it's a little bit of a problem for American public education. So let's talk about what's going into these boxes. Set up some vocab words here. We do have genes. Genes are the full set of instruction that you possess right now, right? The genes are your instruction booklet. We talked about what genes are. Alleles are alternate forms of genes. So if you have brown eyes, you might have 2 copies of instructions for brown eyes, or you might have a copy of instructions for brown eyes, and you might have a copy of instructions for blue eyes that you are not using. So each of those sets of instructions is an allele. Instruction booklet 1, instruction booklet 2 are alleles. Alternate forms of genes that code for the same thing, like eye color, for example. The genotype is the actual genes you have, like having a copy of brown eyes and a copy of blue eyes. That's your genotype. The phenotype is what gets expressed. So if the person has brown eyes, their phenotype is that they have brown eyes. And I tend to use eye color as the example here, but I want you to be aware that this goes for literally everything in your body that is based on proteins. So the chloride ions in your pancreas. You could have one set of instruction for chloride ions, or you could have a different set of instruction for chloride ions. Chloride ion channels is the word I'm looking for. Chloride ion channels in your pancreas. One set of instructions or 2 sets of instructions. They could be the same, they could be different. Those are alleles too. So don't get stuck on the idea that it's just about eye color. It's about every trait, and every trait has a lot to say about your physiology and the functioning of your system. So again, the phenotype is just what gets expressed, but you could have a hidden world of stuff in your genotype that is not getting expressed, especially if you are heterozygous. So that person with brown eyes and 1 copy of brown eye and 1 copy of blue eye, they were heterozygous. They had 1 dominant, brown eyes, and 1 recessive, blue eyes, gene. They had the dominant allele and they had the recessive allele. A different person with brown eyes may have 2 copies of instructions. Both of those instruction booklets are for brown eyes. Brown eyes are dominant, so that person would be homozygous dominant. That means you have 2 dominant alleles. Both of your chromosomes, the one you got from matrilineal and patrilineal sides, are both representing the dominant allele. Another person, let's say with blue eyes, could have 2 recessive alleles. If somebody has blue eyes, that by definition is a recessive trait. You need both of those copies to be the recessive form in order to express that condition. So somebody with blue eyes has 2 copies of the blue eye allele by definition. That would be homozygous recessive. If I was not homozygous recessive, if I had any 1 copy of a dominant allele, my eyes would be that color instead, because a recessive trait can only be expressed if it's fully recessive. Any dominant trait would dominate that characteristic. Let me know if you need clarification on that in the discussion forum. So again, dominant, if you possess 1 or 2, it is dominant and it will be expressed in the phenotype. Recessive only can be shown if both copies of the allele are recessive. And then you do have other patterns of inheritance, incomplete dominance. So for example, hazel eyes, green eyes. We get these little in-between things that don't quite obey the rules. It works for like colors of animals' coats. If you're going to be spotted or mottled or tricolor, those tend to be incomplete dominant traits. Some traits are polygenetic. As I said before, skin color. Many genes determine a person's skin color. We can't say, oh, you have gene X. That means you are this shade on the Pantone color wheel. That doesn't work that way. It's polygenetic. And then occasionally some things are sex-linked, which means they're found specifically on the X or Y chromosome. And the reason we have to specify sex-linked is that the rules are slightly different. The X chromosome is a lot longer than the Y chromosome. It's not that the Y chromosome is Y-shaped and the X chromosome is X-shaped. That is inaccurate. The X chromosome is long and the Y chromosome is simply short. As such, the Y chromosome carries far fewer disorders than the X chromosome. The X chromosome is much more likely to carry disorders. But if you have 2 copies of an X chromosome, that second X chromosome is protective. So if you have 2 copies of the X chromosome and you are considered to be biologically female, you are actually protected from a lot of genetic disorders. Whereas if you have an XY chromosome and there's a disease state on the one X chromosome you have, on your Y chromosome you are unlikely to possess the undiseased state to correct for that. I can discuss that further if you would like. Now again, the X and the Y that was on the first slide, that was for X and Y chromosomes. All other traits, in genetics we usually have some kind of designation, some kind of lettering designation. Capital A, lowercase a, uppercase B, lowercase b. Both of these, in this case, are talking about a heterozygous person. So this person is heterozygous, this person is heterozygous. It looks like we are talking about albinism. Now, albinism is a recessive trait. You need 2 copies of the recessive allele in order to express albinism, being an albino. Neither of these parents are albino. They phenotypically have normal pigmentation of their skin because they both have a dominant allele and their body is taking that dominant information and utilizing that dominant information in the cellular processes. That's what a dominant allele does. It is the one that dominates the system at play. So both parents are heterozygous. They are both phenotypically typical, but they're both carrying the recessive form of albinism, either through random mutation or because somebody in their family is an albino. They were both born with the recessive albino gene. So these 2 people in all of the world managed to find each other. Maybe it's a support group for people who are in the families of people with albinos, or maybe they're closely related, which is actually frequently how this works out. Not all the time. Not all the time. Okay, so 2 parents got together. Both of them have the recessive allele for albinism. So maybe they go in for genetic counseling because they've seen a lot of family members die of skin cancer and they don't really want that for their offspring, and they want to find out their probability of passing this on to their offspring. So their genetic counselor is going to put this into a Punnett square because this is a trait that obeys our rules of dominant and recessive patterns of inheritance. So they say, okay, parent A, you are heterozygous for the condition. Parent B, you are heterozygous for the condition. Let's go ahead and cross multiply these. 25% chance that your child will be homozygous dominant. This person will phenotypically have normal typical skin pigmentation, and they will not even be a carrier for this disorder. Probability here and probability here mean that 2 out of 4, or 50% of offspring, will be just like you, heterozygous for the condition, phenotypically normal skin pigmentation, but they will both be recessive carriers for the disorder, just like you are. And just probability here says that there's a 25% chance that your offspring will express the phenotype of being albino. This person right here, this 25% chance that offspring will be actively albino. Now, that's not a death sentence. This offspring will need to utilize a lot of sunblock and stay in the shade. There may be some other effects. They're on some different organ systems, but it's not a death sentence. They can live a full life, and there's different degrees of albinism as well. And so that's the genetic counseling. So there's a lot of vocab that goes into it that you will be held responsible for. It's not just for disorders. We can use this system for a lot of genetic traits, such as a widow's peak, which I obviously don't have. Quite the opposite. Okay. The widow's peak is a dominant trait, as are attached earlobes. I have detached earlobes. Let's see, some other dominant traits. As I said, brown eyes are dominant, blue eyes are recessive. Dark hair tends to be dominant over light hair, although that's imperfect, so don't go with that one. So with the widow's peak, the dominant form would be capital W, the recessive form would be lowercase w. And once again, we have two heterozygous individuals, two people who have widow's peaks but are both carrying the recessive, no widow's peak situation like I have. And once again, because these are both heterozygous, you come up with the same probability. 1 in 4 chance that their offspring will not have a widow's peak, 50% chance that they'll be heterozygous and be able to pass on a widow's peak, and a 25% chance that they will phenotypically have a widow's peak and be unable to pass on the no widow's peak condition. So that's actually pretty close to our albino chart. Be cool if we could do something other than heterozygous. Let's look at over here. Homozygous dominant person meets up with a homozygous recessive person. In this case, because of these two dominant alleles right here, 100% of offspring are heterozygous. 100% of offspring are going to express the medical condition, the trait that we are testing for. Two homozygous dominant individuals, guess what? 100% chance that their offspring will be homozygous dominant. Everybody is going to have the dominant form, and they will be unable to have offspring with the recessive form. Or give that to their children, I should say. Heterozygous plus homozygous recessive, 50% chance of heterozygous offspring, 50% chance of homozygous recessive. The best way to learn this is to do it. And if I ask you a test question that means that you need to determine probabilities, you really have to write it out and do it. Nope, don't memorize this. This is not for memorization. Even if you think, oh, I know exactly what that's going to look like a Punnett square, I know the exact probabilities off the top of my head, draw it anyway. It costs 30 seconds of your life to draw it, and you're much more likely to get a question like that correct. I see students get it incorrect all the time who had in-class instruction. We did this kind of lecture over and over again, but they just didn't physically write it out. I get them wrong all the time if I don't write them out. I will see, two sets of probabilities, and I'll think, okay, that's obviously going to be 25%, and I'm wrong until I write it out. Write it out. Write it out. So here's an opportunity to write it out. Julie is going to have, and I'll just give this to you, she's got brown eyes, he's got brown eyes. What eye colors will their children potentially have? That's going to be a good exercise for you. I'm not going to do it for you. Write it out. It's the only way. And again, just a reminder that this comes from gametogenesis. This comes from our process of meiosis. Those probabilities come from random assortment. Those probabilities come from, was it this egg or was it this egg? Was it this sperm or was it this sperm? I know that this is just a generalized thing. This isn't, egg and sperm are not coming from the same source. But that's where we get that 50-50 probability that goes into the Punnett square. This is a probability. This is a probability. This is a probability. This is a probability. This is how those probabilities combine. As I mentioned, unattached earlobes. I thought, I guess I was wrong. Attached earlobes are recessive. Unattached earlobes are dominant. I feel like I've learned otherwise before, but we'll go with it. I'm not going to dissect these charts. You can use these charts as your own. I've used them in different scenarios. Yeah, that's great. Let's see if we can find it. Yeah, there it is. I mentioned earlier how one way that two people with albinism in their background may have gotten together is that they might be closely related. Now, this was a royal family that had a lot of recessive disorders. And one of the reasons that they had recessive disorders is that in the ancient royal families, it was considered to taint the bloodline if you married outside of your immediate family. So they had a certain amount of intermarriage between family members, and as a direct result of that, they had a lot of this recessive condition known as hemophilia in their family line. So this right here, if you understand a family tree, means that two family members, in this case it looks like cousins, got married. Now, if just one incidence of cousin marriage is happening in a family line, it tends not to be a big deal. But in something like the royal family, where they may have been related, or previous generations may have been related, it means you're much more likely to carry a recessive disorder that is similar to the recessive disorders being carried by your partner. This happens more than you would think. It happens mostly in insular communities, communities where there's a cultural norm of staying within the community for your marriage partner. This is another reason that biology likes diversity. It's best for you to reproduce with somebody who is basically the opposite of you genetically, because then you are much less likely to pass down recessive traits, or especially in this case, recessive diseases. That's not to say that those cultures are wrong or bad, it's just to say that they are more likely to occur within those cultures. And that's why a lot of insular cultures know that they need to get genetic testing. What else do I want to say about these? I mentioned X-linked before. X-linked can be a very special case, because having two copies of the X chromosome is going to be protective, and typically the healthy form of the X chromosome will be dominant. Whereas with an XY combination, there's no healthy X chromosome to take over that role. So males are much more likely to express diseases when it's an X-linked disease or disorder. That's what's happening with this slide here. Let's see if we can break this down. So this is a carrier female. So phenotypically, she does not have hemophilia. She has the dominant typical form on one of her X chromosomes. 50-50 chance she passes on this chromosome, 50-50 chance she passes on this chromosome. Meanwhile, the male is phenotypically normal and has the typical X chromosome, and of course his other chromosome is a Y. So I put this into a Punnett square, and what we end up with is a healthy female and a healthy female carrier. And we end up with, this should be a healthy male, and 25% chance male with hemophilia, 0% chance that we have a female with hemophilia. And this is typical for a lot of X-linked disorders. This is also why it's much more likely that a male would be colorblind. Red-green colorblind is the most common form of colorblindness, and it does happen much more often in males than in females. It's very rare to get a colorblind female. Essentially, you need both X chromosomes inherited to be the recessive form, which is a challenge. It can happen, it's just very unlikely. It means mom and dad both had to have the colorblind X chromosome passed down. So again, it passes on mostly to sons, very rarely to daughters. Another issue that can happen, this is really, really relevant clinically, is non-disjunction. So I mentioned meiosis 1, meiosis 2. You're going to have homologous chromosomes separated, and you're going to have sister chromatids separated. At any point, meiosis 1 or meiosis 2, you could have a failure of separation. You could have a non-disjunction event. Let's see, make sure during the first division, in which case one gamete is going to get both copies and one gamete is going to get 0 copies. Oh, sorry, I did this wrong. No, this is very poorly illustrated. Okay, let's try that again. First division, one gamete gets both copies, one gamete gets no copies. That means you end up with 2 daughter cells with an extra copy and 2 daughter cells with no copies. Second division means that the sister chromatids didn't separate. You get one gamete with 2 sister chromatids, one gamete with no sister chromatids. So that's going to mean that you end up with an abnormal number of chromosomes. Now this is only demonstrating one set of chromosomes. Let's say this is autosome 22. So let's say autosome 22, or we could do autosome 21, make your life a little easier. Let's say this is autosome 21. Let's say that this is an egg cell, sure, why not? And it meets a typical sperm cell that has only one copy of the autosome numbered 21. That means that this gamete is going to end up as a zygote with 3 copies of autosome number 21. Now we're only following 21, we're not following 1, 2, 3, 4, 5, we're not following 23, which is XY. The rest of those could be totally normal in this scenario. Those could be typical. It is just this autosome 21 that we are looking at, that you ended up with 3 copies with. That's actually called trisomy 21, and that's what Down syndrome is. So Down syndrome is just simply an extra 21st autosome, and it's just a result of non-disjunction. Instead of separating and going to the 2 different daughter cells, they just stayed together. There was just a failure of separation. That can actually happen to any autosome. It could happen to sex chromosomes. It could happen in meiosis 1, as you can see. It could happen in meiosis 2, as you can see. The outcome is similar. We still get an extra copy either way, or a lack of copy. If the sperm met this egg, we would end up with 1 copy of autosome 21. And my understanding is that is not compatible with life, which is to say that would lead to a miscarriage, which is a big scary word for a lot of people, but is unbelievably common. We actually think it's as much as 25% within the first 12 weeks of pregnancy will end in miscarriage. So unbelievably common in any culture in human history. But a lot of women are not taught, a lot of people are not taught that this is very, very common to have a miscarriage in the first 12 weeks. And a lot of people are going around feeling a lot of shame for their miscarriage, when it really is about chromosomal differences that mean that it's not compatible with life. There's very, very rarely is it the fault of a parent. It's almost impossible to cause a miscarriage willfully, without some very, very complicated drugs involved. So again, you have these different scenarios. You can end up with a trisomy, which is 3 copies of any given chromosome. You can end up with a monosomy, which is only 1 copy of any given autosome or chromosome. And we can detect that via karyotyping. So this is going to be like a prenatal test that we can do. We actually have, this is very much computerized today. This used to be something that had to be done more manually. But what we can do is we can take, once they're condensed into chromosomes, we can take a sample cell, and we can lay out all of these chromosomes. Again, we have computer programs that can assign, okay, the computer program found the first and second copy of autosome 1, the first and second copy of autosome 2, first and second copy of autosome 3, and so on and so forth. And down here, they've actually only illustrated XX. They have not illustrated an XY. So I might go and find that picture for you. All right, I just added the slide really quick just to show you that I was telling the truth. It's not that it's X-shaped or Y-shaped. It's that the Y chromosome is simply shorter. And that means statistically it's less likely to carry a disorder on it. It's just smaller. It's just less. Having less chromosomes is not worse. We have less DNA than, I think, a banana, if I remember correctly. It's not about quantity of genetic material. It's about how it's being used, so to speak. So again, this is the one they gave you for karyotyping. That's karyotyping an XX individual. This is karyotyping an XY individual. And the only difference should be this segment right here. And again, there can be non-disjunction of the X and Y chromosomes as well. So we can end up with XXY, XYY. X monosomy is a possibility as well. You could have just an X without a Y, or another X. That's called Turner's. So karyotyping can inform people of what their chromosomes look like if there have been any non-disjunction events, and then counseling can occur accordingly. And we're not going to get into the ethics of that today. People have different belief systems about trisomy 21 and what is an appropriate response to that. There actually is a trisomy 21, so they actually, yep, there they go. So plus 21 right there. They have 47 chromosomes total. This is a female with trisomy 21, and that's what this genetic test gets us, is that piece of information, and then counseling can go from there. All right, let me know if there are any questions in the discussion forum.